Question: how do we know that our cells are supposed to move to the infection site when we get sick? Thankfully, we have something in our bodies called chemokines. Let's talk about them. Essentially, chemokines are signaling proteins secreted by cells of our immune system that stimulate the movement of other cells throughout our bodies to the infection site when we get sick. Chemokines are a type of cytokine that induce chemotaxis. Chemotaxis is basically a big word for the patrol of our immune system's cell movement throughout our body and throughout our tissues. But how does this work? The way that chemokines do this is by establishing a chemotactic gradient, which is sensed by immune cells that have receptors for the chemokines. So for example, let's say we have a cell that is secreting a chemokine due to an infection somewhere in the body. The immune cell sensing that chemokine will move up its concentration gradient towards wherever the infection is in the body. Because there are so many different infections that we can have in our bodies, we'll need different chemokines to interact with the specific infections. However, my interest lies within the chemokine CC motif ligand 2, or CCL2. CCL2 is a small cytokine that belongs to the chemokine family. CCL2 recruits monocytes, memory cells, and dendrites to sites of inflammation as an immune response that works primarily in our central nervous system with our brain and our spinal cord. We know that the hypothalamus plays a crucial role in the secretion of hormones, especially during the time of puberty. The hypothalamus specifically releases gonadotropin-releasing hormone, oxytocin, and prolactin hormone, which affect the reproductive systems of the male and female bodies. And here lies where my research begins. If chemokines are active in our central nervous system, especially within the brain, and the brain has these hormone-secreting properties, then what better way to see CCL2's effect on our bodies than by a visual test such as analyzing the rate of puberty. Currently, I'm working with my professor to see whether the implementation of an extra CCL2 gene will have an effect on the rate of puberty within mice. We are currently looking at wild-type mice with a normal expression of CCL2 and transgenic mice with an overexpression of CCL2. Here I am checking for vaginal opening in female mice and preputial separation in male mice. These factors will determine when the mice undergo puberty. We will then compare the times of puberty in wild-type and transgenic mice. We now know more about immune response in our bodies with chemokines, and hopefully we'll know more in the near future about CCL2 and how it can possibly benefit immune deficiencies in medicine.